Yeah.
Okay.
Okay, are you ready to start?
Yep, I am whenever. Just tell me when.
Yeah, please start.
Okay, first of all, good morning, good afternoon, good evening. I don't know where my audience is from, so yeah, good morning, good afternoon, good evening to everyone. Like Irina introduced, I'm Tek Malla from University of Wisconsin-Milwaukee, and currently I'm a postdoc at the laboratory of Marius Schmidt, who you may have seen earlier. He's in urgent travel, and he's listening to us from his car right now. So I would like to start by thanking Irina for the invitation to give this talk. The talk today is about ID-29 at the European Synchrotron Radiation Facility with the new upgrade called the Extremely Brilliant Source upgrade. But I'm not talking about the progress or the capability of the beamline, right? I'm not the right person. Today, I'll just share my experience conducting our experiment at this beamline because I have had some pleasant experiences. You see, I started doing my doing crystallography at the free electron lasers, and so far I think I have been to 15 XFEL experiments. But this free electron lasers experiment were never a walk in the park. There's always something that went wrong or that goes wrong. For example, sample gets clogged. If the sample's not clogging, the beam goes down. If the beam is right, the geometry is not great. If the geometry is right, the detector is not working right. Something always goes wrong. And then the 4 days of beamline is actually very hectic, right? And this is not to say we do not get good results. In fact, all the success that I have achieved so far were all thanks to our experiments at the free electron lasers. Anyway, let me share some experiences and present the results and discussion as they come along. So as you're all aware, the ESRF went through an upgrade and then came back operation back again last year. This new upgrade is called the Extremely Brilliant Source upgrade. And then after this upgrade, they built a new beamline, and this is actually a flagship beamline at the ID-29, and this beamline is entirely dedicated for room temperature serial crystallography. Like they built this beamline in the laboratory from ground up. So they installed all the latest, fanciest, the best detector they could find, this Jungfrau OM detector, and a diffractometer where they can put both fixed target or foils, or use what is this high viscosity extrusion or tape drive for serial crystallography. And then, a new modern lab space where you can prepare your sample or characterize your crystals and whatnot. So when this beamline came into operation, the beamline scientists, they contacted us, because Marius's laboratory has had some success performing time-resolved experiments. So they reached out to us and asked, hey, Marius, we have a new beamline for time-resolved serial crystallography. Would you like to perform your experiments and maybe commission this beamline? So they reached out to us, and then we were immediately excited. This is a huge opportunity for us and everyone. There's a new synchrotron beamline, that is redefining what serial crystallography is, because free electron lasers is too expensive and there's not so many of them. So anyway, we accepted their request, but deep down, we knew what we had to do. It's like we were supposed to use their beamline and then give a review to everyone in the community. Basically, we were expected to answer few questions. For example, number one, can we get good resolution? Right around in 2023, beginning of 2023, I went to some conferences, and heard some PIs telling the resolution at ID-29 is not good enough, right? It cannot match the resolution of XFELs and whatnot. So we were also skeptic at first, right? Like, does it get good resolution? If the resolution is not good enough.
I'm sorry, can I interrupt? Can you explain for people not in the field how you define the resolution in this case?
Okay, yeah, I guess. Good resolution, yes, can be pretty vague, but we were expecting optimally around 2 angstrom. So anything better than 2 angstrom is really high resolution. 2 to 3 is medium, and then less than 3, we would say it's not good enough. I mean, also some results where we have result at only 3.3 angstrom. but on the best case, we're hoping to get resolution beyond 2 angstrom. So that is the first question we needed to answer.
But I'm sorry, but my understanding was it's usually limited by the crystal.
That is true, but then it is also limited by the flux. If there is less flux, then the crystal does not diffract well, and then it also depends on the detector. If the detector cannot catch a weak reflection, there are so many different factors that affect the resolution. But yes, crystal is one of the biggest factor. If you have a really good diffracting crystals that diffract to high resolution, you can get good resolution. So the second question is, since ID-29 was purpose-built for serial and time-resolved crystallography, as such, does it have any competitive advantage over beamline? And then finally, is time-resolved crystallography really feasible? Like feasible as in routinely, like all other synchrotron experiments in the past, right? So to find out answers to each of these questions, we tested the capability of this beamline under different scenarios using various microcrystal from different proteins. So our first protein is photoactive alloprotein, and this is our model system for testing new beamline. I feel like our laboratory is the only laboratory that does not use lysozyme, because other than us, everybody who does something new for the first time, they always use lysozyme. But anyway, photoactive alloprotein is a small blue light photoreceptor whose photocycle in crystal is identical. It's slightly different from solution, but it's mostly identical to that of solution, and it is also reversible in the crystal itself. Both the proteins and crystals are incredibly stable. They last for years and diffract to highest resolutions. So it has been used as a model system for studying ultrafast dynamics with time-resolved crystallography. For example, if you look at the literature, it was used to establish millisecond time-resolved Dirac crystallography at synchrotrons back in 1997. And then the time resolution got better with nanosecond in 2005, picosecond in 2012. And then when free electron lasers with femtosecond time resolution capability were introduced, once again, PYP was used as the model system, both at European XFEL and LCLS, to perform picosecond and femtosecond time resolved crystallography. So we used fixed target. At the moment, they had a fixed target and tape trap available for serial crystallography. So we used fixed target first. So the way this fixed target works is you have these 2 mylar films. So there is, you take a mylar film and draw few microliter of your microcrystal slurry. I think we put around 2.5 to 3 microliters. And then you sandwich the sample with another mylar film and then put it in front of the X-ray beam. And then the X-ray beam scan this predetermined spots. It performs a raster scan. For example, it illuminates here and then here in a serial fashion. Right, and then these are our PYP crystals. You can see how clean and beautiful with sharp edges they are, and then they diffract really, really well. So this is the electron density map we obtained from PYP. The protein is well resolved, and when you zoom in on the inside, you can distinguish the individual side chains, right? Each and every side chain is well resolved, and this is the chromophore for the protein. And if you focus on this chromophore, like the chromophore is also well resolved, you can see the donor set hole in this, what is this, 1 sigma map, and then it translates to 1.8 resolution, 1.8 angstrom resolution, right? So this is a really high resolution. But if there is some veterans out there who have had experience with PYP in the past, you will be thinking, well, PYP is a really high symmetry, and you might be saying, Tek, if you did not get that high resolution with PYP, it would be pretty disappointing. And I would honestly agree with you. This is our thought too. Yes, we get really high resolution, but we also know that PYP has a hexagonal symmetry. So, even with less diffraction pattern, we get high resolution. That's known. So then we used another crystal that we did not mind spending a little. So for this, we used what is this main protease of coronavirus 2, and it has a C2 symmetry. So it's a 30 kilodalton protein with one subunit in the asymmetric unit, and the next subunit or the dimer is related by the C2 symmetry, right? And then with this, we managed to get 2.3 angstrom resolution, which is comparable to what we obtained at both LCLS and the European XFEL. And if you look at the map and model, you can once again see all the individual side chains are well resolved, right? And these are the microcrystals that we used. So then these two examples demonstrate that the resolution is really not an issue at the beamline. Yes, there are factors that might affect the resolution you can get, but if you have good crystals, then the beamline is well suited for really high resolution experiments. In fact, we maxed out the resolution with PYP. So earlier I said detector plays a factor. So if the detector, the closer the detector is to the X-ray interaction region, the better the resolution you can get. So with PYP, the resolution at the corner of the detector was 1.8 angstrom, and we maxed that out. And at that point, we could not go any closer because, you know, the beamline, there were so many other stuff that the detector could not be brought any closer. And then this experiment was performed in July last year, but I was at ESRF last month for a conference. And then there they said they are able to move the detector closer. And I think they say now you can measure up to 1.6 or 1.45. I forgot which one it is, but yes, you can obtain even better resolution now. So now to the next question, whether this beamline has some trick of its sleeve. For that, oh, I forgot to say that we answered the first one. So for the next question, we use this protein called cytochrome C nitrite reductase. It's a decaheme protein. That means it's a dimer with 5 hemes in each monomer, right? And this so the hemes are shown in here, and this is the cartoon representation, and this is how they are arranged. So the electron. Before that, this enzyme, it is responsible for the 6 electron reduction of nitrite to ammonia. In addition, it can also reduce nitric oxide, and then what it is, hydroxylamine. Right, to do that, the electron enters to the enzyme through this heme site on each of this subunit, and then this heme on the top here is where the reaction happens. But all these hemes are used to transport the electrons around. And these two hemes at the dimer interface are so close together that the electrons can even jump around from one subunit to another subunit. That is pretty cool. And then this the study in this enzyme is so important because, you know, it plays a very crucial role in the nitrogen fixation or nitrogen cycle. This is a cool protein, but it's not easy to work with. In fact, nothing about this protein is easy. For one, it's expressed natively. There is no overexpression system for it to be expressed in E. coli, at least not with the wild-type CCNIR that we have from this bacteria. I forgot the name at the moment. There is no overexpression system, so the yield is only 0.5 to 3 milligrams per liter. Like, let that sink in for a moment. 0.5 milligram per liter. When I grow cells, I grow in 1 liter flask, and the laboratory next to us who does what is this optical tweezer experiment with protein, they say, this is a huge amount of cells I'm growing to make protein. When I grow in 1 liter flask. Victoria, the student who purified this protein, grows her cells in 50-liter container. 50, not 15, 50-liter container. It takes 7 to 9 days to grow the cells. And then after 6 or so different purification steps, and then more than 2 weeks of laborious work, you obtain only 30 to 150 milligrams worth of protein, right? The yield is abysmally low. And then later you will see that more than half is again lost to crystallization. So we had a crystal screening beam time at the European XFEL, and then we made two crystals with two different conditions. So I'm saying here condition 1 and condition 2. So they are crystallized with the same batch of purified protein, right? We mixed one mother liquor in condition 1 and the different mother liquor in condition 2, and then we end up with these two crystals. So with condition 1, visually, they look great. But we put them in front of the X-ray, they did not diffract at all. We tried to characterize them with electron diffraction. We did sonic and stuff, not crystals, but they look like crystals.
So you showed the crystal structure, and now you say that nothing diffracts. So is it the different protein, or what's the goal here?
Crystal structure of this CCNIR, this protein?
Yeah, so what is here?
Yeah, so I will come to this later. So condition 1 did not diffract, but I'm coming to condition 2 and then another condition. So condition 1 did not diffract at all, although it looks good. But then condition 2, although it looks dirty, there are some crystals in here, and these crystals diffracted exceptionally well, right? So as a crystal screening beam time, it was a success because, we got some diffraction. But for a student who was hoping to get results on time and put it in a thesis and graduate, it was a sad outcome. But what we learned from this experiment is by looking at these two conditions, because they were prepared from the same batch, the protein is still not pure enough because you see still a lot of some particle. So what we did was we fished out this really nice looking crystal-like particles from condition 1 and recrystallized with the condition 2. And then it gave us this crystal, which are super clear and then super nice, and then they also diffract very well. And then we use these crystals at the ID-29 beamline. So at the end, after all that fiasco and recrystallizing and losing half the protein in the process, we were left with only 10 to 15 microliter of protein. And you can imagine 10 to 15 microliter for serial crystallography. It's pushing really hard. Like it's not ideal. You need tons and tons of protein. But fortunately, the fixed target method, they do not require a lot of protein. So with those 10 or 15 microliter of sample, we collected around 2,500 diffraction pattern. For example, with other PYP and whatnot, we obtained around 50,000, 60,000 diffraction patterns. And then this resulted in this 3.3 angstrom resolution. The side chains are kind of well resolved. And then if we look at the heme cofactor, the hemes are also well resolved. And then at least when we are looking at it, we did not see any sign of radiation damage, right? And if we compare our structure, this green is our current structure from the serial synchrotron crystallography, and we compare with the cryostructure obtained in the past, which is shown in this blue. You see first the room temperature structure, one of the unit cell parameters increases by 5 angstrom. And then the another difference is that next to one of the monomers, there is a PEG molecule. And if we zoom in here, so this is the PEG molecule, and I'm showing the FO minus FC difference map. And then there are this, there's this electron density that traverses the entire unit cell, and then we can fit a PEG molecule. At the moment, there's only 10 monomers of PEG that can fit in there, and the rest is related by the symmetry, but then it just keeps going. And if we look at the crystal packing, so this is the unit cell again, and then this is the electron density for PEG. I only shown 6 of them, but you can imagine like inside each and every gap in here is a PEG molecule. So then it traverses the width of unit cell and possibly the entire crystal. And then what I assume is happening is, this we were using PEG 4000. It's a long strand, so it's like a thread that sews and weaves around the protein and then brings them into stability. To me, as a crystallographer, to see the PEG doing that to molecules and the crystal, that was very, very cool. But I cannot hide the fact that this is still only 3.3 angstrom, right? Like I said earlier, anything below 3 is not that great. So we need it to get better. So was there any room for improvement? Yes, the answer is absolutely yes. So see, this is the chip. And then when we were collecting diffraction pattern, we were collecting diffraction pattern from this central predefined area, right? So as we do not, because we are absolutely sure that the sample would be here, but we were not sure if the sample would reach the corner. So we were excluding these sites. But the fact is sample zips and sample extends beyond this predetermined area. So we could have gotten further and increased the scan area, and then that way we could have gotten more diffraction pattern. But the even more important thing that we missed is the spacing between these scan points is 20 micrometer, 20 micrometer. But the size of the beam is only 4 micrometer. In fact, it is 4 micrometer by 2 micrometer. So even if we had gotten 10 micrometer, there still wouldn't be any contamination from the X-ray beam. So if we decreased our scanning points by half, this distance by half, we would have ended up with 4 times more diffraction patterns. So instead of 2,500 diffraction pattern, we could have ended up with 20,000 diffraction pattern. And that would have been enough for us to get sub 2 angstrom resolution. And because the beam is 4 by 2 micrometer, it's a rectangular beam. So on the side where it's only 2 micrometer, we can even go closer and decrease the scan distance to 5 micrometers, right? And then if we had done that, we would have ended up with 40,000 diffraction pattern. So 40,000 diffraction pattern for the orthorhombic crystal, we for sure would have gotten a resolution better than 2 angstrom, because this crystal, with this crystal, we already have a resolution of 1.6 angstrom from home X-ray source. So hindsight is always 2020. We did not think of it during the beam time. We had very limited sample, and now we do not have any more sample at the moment to repeat this experiment and then put these things into consideration. So if there's anyone out there who is considering using fixed target or similar setup, please remember this in mind, because if your sample is really precious, you might get more out of it than what is already pre-set up at the beam line. Oh, here's the slide where I was saying you can go further with these scanning grid points. So why did I focus so much with this CCNIR? Because CCNIR is a really special protein. Like I said, it requires 6 electron for it to function, and this 6 electron reduction reduction current is actually measurable. In fact, in the past, scientists have already used this as an electrode to measure the current, right? And since this enzyme is very specific to nitrite, it has wide-ranging application in wastewater treatment, agriculture, or even greenhouse emission. But you must have already realized it requires an electron donor. Without electron, it does not perform, or it does not react. So there is an active research going on in synthetic chemistry where people are trying to develop novel electron donors. But in order to design such thing, first we need to comprehend or understand the electron pathways and the mechanism of this enzyme. And for this, of course, one thing we can do is time-resolved crystallography. For example, we can perform a simple mixing experiment where we have CCNIR microcrystals in one reservoir and nitrite solution in the other reservoir. We simply mix them, that will trigger the reaction, and then we inject them into X-rays. So far, we have been using really precise microfluidics mixtures for our time-resolved experiments, because we wanted to go as fast as possible in the 1 millisecond or sub-millisecond regime. But this reaction is not that fast. So with that, what I'm saying is we do not need a very precise microfluidics, and we do not need X-cells per se. So this experiment can easily be performed at synchrotrons. So now, was there any advantage of ID-29? This is a difficult question to answer, but I incline with yes. The sort of polychromatic beam, 1%, I think, which is 10% higher than crystal, meant we have a complete data set with only 2,500 diffraction patterns. At the moment, this is speculative. I'm not quite sure.
Chromatic beam, so it's everything is Lowy diffraction, you want to say?
It's not Lowy. I think it's quasi-monochromatic. They do not advertise this as a Lowy, but I just assume that because we have 1% bandwidth, we might have we were able to complete data set. But at the moment, this is my speculation. I'm not quite sure because this beamline came into operation very recently and more work needs to be done. And then with this
It
protein,
would be helpful to have a table with like beamline characteristics because like otherwise it's really hard to follow.
Yulia, can I pitch in? Yulia, first slide was a beamline layout. Perhaps you didn't pay attention, and it was a table with the characteristics and beam band pass. So it is 1%, which is I think multi-layer, right? So it's a quasi-polychromatic and quasi-monochromatic. So yeah, that's what I would like to say.
Yeah, I had a table, but I did not dwell on the table. I apologize for that. But yes, I had a table with the beam characteristics at the beginning of the slide. But anyway, the point I was trying to make is I do not know if this is really true, but we have the means to make sure. With this protein, cysteine-IR in particular, we do not have data set at European XFEL or another free electron laser to make a direct comparison. But with PYP, we have data set from both ID-29 and at European XFEL, and then we can do the comparison to see if that is true. But at the moment, we have not done that because we are busy with other stuff at the moment. But that is something I think we might do in the future. But another advantage was that, this beamline operates at room temperature. There is no enclosure or anything. So you can quickly swap out injector for chip or tape drive. So when we were performing this experiment at European XFEL, we were using a liquid jet. And then we realized, okay, liquid jet clocks with this PEG 4000. Can we use chip? The answer is yes, but it would have taken time, and then we were only given 3 hours. So during that time, we could not have changed to a different sample delivery method and gotten our data set. Whereas at ID-29, because they're part of everything, it's really easy to swap out sample delivery method, and then you can start collecting right away. And I think that's the big advantage. And at the end, I'm just happy that we have a structure of a complex protein. We have the structure, the paper is ready for submission, and a student graduated on time. And then asking for anything more at this point would be being too greedy, in my opinion. So then the final question is time visual crystallography possible? For this, we used phytochrome. Right? Phytochromes are red light photoreceptors found in plants, fungi, and bacteria. And in plants, they regulate essential physiological processes like seed germination, shadow avoidance, chlorophyll production. But they are also present in non-photosynthetic bacteria. And the question is, what are they doing in non-photosynthetic bacteria? And lately, these phytochromes have been used as fluorescent markers for non-invasive tissue imaging, because phytochromes, they fluoresce in the far red region, so they can penetrate deeper into the mammal cells. So they have been used as fluorescent imaging in mammalian cells and mice. And also, they are being used as optogenetic tools because with phytochrome, there's this response regulator associated to it, which regulates the gene expression. So the gene expression is essentially can be regulated by light, which is sensed with this phytochrome. So yeah, there's increasing use in optogenetics as well. So this is the structure of phytochrome. The phytochrome has 2 distinct states, PR state and PFR state. So the ground state is the PR state. So in the ground state, the phytochrome absorbs this 640 nanometer light and then goes to this PFR state. And then from PFR state, the protein can go back to PR state by observing this far red 750 nanometer light, or in absence of light, it can go back itself, but much slowly through thermal reversion. And then in this protein, somewhere in here in this core, is a chromophore called biliverdin. The structure is shown in here. It's a tetrapyral structure with rings A, B, C, and D. And then when the protein goes from PR state to PFR state, this ring D isomerizes between this 15-16 double bond. So right now it's in cis or G configuration, and upon photoactivation, it rotates and then goes to E or trans isomerization. So that is one of the characteristics. The other characteristic is this part of the protein, which we call sensory tongue. So it's this sensory tongue is part of this domain on top, and it goes down and it probes the conformation of the chromophore. And then in PR state, this is a parallel beta state. And in PFR state, this is a split alpha helix, right? To me, this is just remarkable, right? Because I have heard of a beta sheet breaking structure and becoming a flexible loop, or an alpha helix losing its helical shape, but a beta sheet dismantling itself and then reorganizing itself into an alpha helix and vice versa, that is new. And the thing is, nobody seemed to explain why this happens. There's almost, 15 to 20 years of research in phytochrome structural research, but nobody seemed to know how it happens. They always state in every publication that the protein does this, but how does it do it? Nobody has the answer. Anyway, so to understand how this photosensor works, we need to do time resolve experiment. And so we did using this tape drive. So we simply swapped out this fixed target and then installed this tape drive and started collecting right away. So how it works is, this is the tape that moves this way, and then we apply sample, and the sample, it gets stuck in the tape, and it's delivered slowly into this X-ray interaction region, which is this red line here. But right before this X-ray interaction region, we are signing this spot with a red diode, right? So then this red light triggers the reaction in this microcrystal, and then any conformational changes are probed by the X-rays. So right now it's set up such that we obtain 700 millisecond palm probe time delay.
Sorry, another question. Is this a home build or is it commercial?
This is not commercial at the moment. I think there is a group, I don't know which group, that made the tape drive and provided to this, but this group is not the only one that's making this tape drive. I think the Lawrence Berkeley National Lab is also making this tape drive. But as far as I'm aware, I do not think this is commercial, but maybe it will be commercial, because after we had success with this tape drive, I know a lot of people asking if they can use tape drive for their experiments as well.
But it's available at the beamline, so.
It is available at the European Excel beam, and that is correct. And then if you rotate this setup for this tape drive, you can see how conceptually similar it is to this conventional liquid-based injection method. So the crystals are simply delivered from up to downstream, and then right before the X-rays, it is pumped by some laser of appropriate wavelength. So this is the difference map that we obtained 700 millisecond after.
So might I interrupt and ask a question about this layout?
Yes,
when you have it depicted, you've got this 100 micron diameter beam that's illuminating. The tape is moving at a constant velocity.
Yep.
And so is that light on continuously?
Yes, it's a continuous LED. Yep.
So then what goes into the path of the X-rays, some of what goes into the X-rays will have been illuminated 200 milliseconds before.
Yep.
And 200. So it's smearing out. So when you say 700 milliseconds pump probe,
it's from here to here. Yes, we are aware of that, but in the past we used precise lasers, but the laser was not installed when we did this experiment. So we had to make do with this. So when we said 700 millisecond, it's from this first point of illumination to until it is probed by the X-rays. So yes, there is some smearing or some protein have been exposed for longer.
Oh, okay.
Oh.
So to be consistent with your 700 milliseconds, that blue dot should be further to the left of that. Right now, it's right at the edge of the intersection. So some molecules will be near 0 time. And
Oh,
some
no. I think I drew this wrong. There is some a gap between this LED exposure and the X-ray interaction.
Right, that's why the blue dot should be to the left by several widths in order to be really at 700 milliseconds. That was inconsistent what you said in the title and what I saw in the picture. So thanks for explaining that.
Yeah, no problem. Yeah, okay. I guess I was not I did not draw this schematic properly, but yeah, there is some gap between this laser LED spot and the X-rays. So this is the difference map that we obtain. Right, in this blue here is the resting structure or the dark structure, and the orange here is the 700 millisecond activated structure. So two things I want to point out. Number one, all this difference map were kind of local, arranged around chromophore. So then we can this D ring is rotated so that the nitrogen points downward and is in E configuration. But even more important is the condition with this water. So this is a conserved water. It's called pyral water because it stabilizes this chromophore by making hydrogen bond with all these nitrogens with pyral rings A, B, and C. It's called a pyral water, and it's conserved in all phytochromes. So this water gets photo-ejected. In our ultra-fast experiments at 5 nanoseconds, we see that this pyral water getting ejected from here. But back then, we did not know where it went, only the fact that it went. And then when in PFR state, this water comes back. Now at 700 millisecond time point, we see this region in here, right below this D ring, where the pyral water likely relocates. Right, so the pyral water moves from here to here, and this can already answer a lot of questions. And this here is just an extrapolated map showing a clear density where the water relocates. So it's right below this D ring. It will come in handy in the next slide. So this experiment was conducted in July 2023. So what is this? And then Marius wrote ID-29 is a TRX time resolved crystallography beamline, and everybody was excited because this has not been successful before. Nobody has been successful with time resolved crystallography there. So the beamline scientists were celebrating, we were celebrating. But when the beamline scientist asked us if they can show this picture and this result at IUCR conference, in August, a month later, if they asked us if they wanted to share this at the IUCR conference, we declined, right? It was an opportunity for them to really brag about their beamline in the biggest crystallography conference, but they were not able to. Because the thing is, we ourselves were not absolutely sure if what we are seeing is true. Sure, the signals are in the right place, they make chemical sense, but we're using very weak LED, right? So we did not trust the photoactivation. For reference, when we did nanosecond experiments with at free electron lasers, we used powerful, high precise lasers, pulsed lasers. But this LED is untested. We were not sure, so we refused, right? So a month later, again in September, we repeated this experiment. In fact, in September, me and Marius, my boss, we were at the LCLS users conference giving our talks, and then we left the conference in the middle of conference, so we could go travel to France, repeat this experiment, because it was important even for ourselves that, if we report something, first we need to be sure that what we're doing is right. So we went back, performed the experiment, repeated everything, but used a slightly different time point, 700 millisecond, but in the logarithmic scale, 700 and 800 millisecond are quite same, just to remove any any inconsistency. If it's the same time point, if you see the same result, maybe there's something wrong. So we selected a different time point, and if we see the similar signal, then our experiment worked. So we repeated with 800 millisecond, and we were relieved when we saw the exact same signal at 800 millisecond time point. And only then we trusted ourselves that what we have here is a legitimate signal, and then we can use this result to talk about biology.
Okay, Tek, may I ask you a question? This is
Yeah.
Irina. You know, my question is, you said that previously people cannot get time resolved data at this beamline, and now you were able to repeat this experiment successfully two times in a row. So what exactly have changed at this beamline? Right, there are no miracles. Oh, perhaps they changed configuration. Something else was done differently.
No, I think it's just that we have better diffracting crystal, and the crystal that reacts to light right away. Because other people, I think, I don't want to call out names, but other people are trying to use photo-cased compounds and stuff like that, and they were not successful. Number one, the reason being a nanosecond laser was not set up, and then nobody really trusted that this LED would work. In fact, this LED, although I am showing it as some sophisticated LED, I think it was just something just a little powerful than a laser pointer that we use for presentation. So it was not a high-powered LED at all. I forgot the exact power of the LED, but yeah, it was not that powerful at all. And that is the reason other people did not even bother using this LED. But we were interested, you know, if it is there, the laser was fortunately the right wavelength. We're like, let's just do it and see what happens. And then fortunately it worked. So we were excited at first, but then we needed to repeat it to make sure that it works.
Okay.
So this was September last year, but again, I was there a month ago, and then they said a laser is inside the beamline. It just needs to be installed, and I think they are installing and performing some of the first experiment as we speak right now.
Okay, thanks.
Yep. So now we go back to this phytochrome and its mechanism. Here and down here, I have this what is the structure of phytochrome, and then it is a video that shows this transformation from this beta sheet to alpha helix. Right, this is obtained from a cryo map, so interpolation with 3 different structures. Anyway, it shows this transformation of this sensory tongue from beta sheet to alpha helix. And then this on the top left here is the sensory tongue chromophore in PR state, resting state. And then in the middle here is the PFR state, fully activated state. And then on the top right here is the overlay of PR, the ground state, it's shown in white, and the activated is shown in color. So this structure and this structure, they are both same. I'm just overlaying it. So in order to explain how this transformation occurs, I am calling it the Dover-Lockdoor model. It's not very creative, but hear me out. So this pink one is the chromophore, and this strand down here is actually the sensory tongue that's here that probes the chromophore. In fact, there are these 5 residues, and these are conserved across all phytochromes. They make direct contact with the chromophore. So in this PR state, this arginine 457 makes a salt bridge with this aspartate, right? And this is a lock in PR state. So this sensory tongue gets locked as a beta sheet in PR state by this strong salt bridge. So then in PFR state, this arginine 459 is kicked away. Instead, this serine 459 comes inward and makes hydrogen bond with this aspartate. And this is the second lock that locks the configuration of both the chromophore and the sensory tongue in PFR state. So if my hypothesis is true, if you remove this arginine 459, then the back reversion to PR should fail. And yes, it has been experimentally verified with the spectroscopy where they mutated this arginine 457, and then protein has a hard time coming back to PR state because this lock does not exist anymore. Similarly, if this serine is not there, then the protein should not be stable in PFR state because this serine locks the position in PFR state. And once again, that has been experimentally verified with this spectroscopy result, right? So PFR lock is missing. That's fine. But then what about this transformation? For example, there's this phenylalanine 460. In the same experiment, they removed phenylalanine 460, and once again, the protein does not transform to PFR. So what's happening there? Because, if you see here or here, phenylalanine does not make any hydrogen bonding or salt bridge or any other interaction with the aspartate that is directly below this chromophore. And this phenylalanine is what I am calling the handle of the door, the driving force. Right? So if you look at this overlay, this phenylalanine is buried deep inside this buried inside this hydrophobic environment, this hydrophobic side chain of phenylalanine in this white structure, which is the ground state. But in PFR state, in the light activated state, the nitrogen of this ring D comes down, the aspartate moves up, and then this tyrosine, they all create this hydrophilic environment in here. And if you remember from previous slide, this region is exactly where the pyrrole water relocated. So my assumption is that the pyrrole water, the relocation of pyrrole water to this position mediates the congregation of all these hydrophilic components in here, and then the robust hydrophilicity kicks this phenylalanine away. And then the rotation of this phenylalanine triggers the transformation of this straight beta sheet into this helical alpha helix. Now, this is again speculative. We need more time points, number one, and then we need a better reaction triggering mechanism like a nanosecond laser so we can be absolutely sure. But at the moment, I'm convinced that this is how it works. But because I have never encountered this protein changing from beta sheet to alpha helix before, so if there's anyone here in the audience who has seen something like this before, please let me know, because I really want to know. To me, this is such an enigmatic process. I've never seen this, and I find this very, very fascinating. So as a conclusion, so did we perform time visual crystallography? I believe that we did. Not only that, we were able to probe this meaningful time point. For example, at XFELS, people do not prioritize this longer 700 millisecond time points, right? Because XFELS are best suited for ultra-fast experiments and picosecond or femtosecond time scales. On the other hand, you can do cryo-EM. I mean, I have done cryo-EM, but cryo-EM cannot resolve these subtle differences or subtle conformational changes or the motion of these residues. So as shown here, this serial synchrotron crystallography filled in this essential knowledge gap left by both TRSFX and cryo-EM. And this was all possible because of the upgrade at ESRF, but we know that APS is also going such upgrade, and then there are several other synchrotron beamlines around the world that are going through upgrades, and then I think in the future more and more synchrotrons are poised to open serial crystallography to wider scientific community. And I think the future of synchrotron is extremely brilliant. And with that, that is the end of my talk. I would first like to thank BioXFEL that funded my PhD, or that is still funding my postdoc. And then big thanks to Northeastern University, because they are longtime collaborators and they provided samples for phytochrome. And then the ESRF, of course, where we got the invitation to perform our experiments. And I'd like to thank all of you for listening. Thank you.